Hey everyone! In this video, I'm going to be talking about the process of viral replication. So when discussing viral replication, there are 7 steps. The first is attachment. And in this step, there are molecules on the virus that will attach to the cell that it's trying to infect. The next step is penetration. So obviously the virus can't do anything to the cell if it's on the outside of it, so it has to get in somehow, and that's what occurs in this step. The next step is uncoating. So viruses can be enveloped or non-enveloped, and that means they can either have basically a shell of phospholipids, sort of like the cell membrane layer, and that can be surrounding the genetic material in the virus. And so if viruses do have one of those shells or envelopes, here I'll write that here, envelope, and I'll put question mark, does it have one or not? If they have an envelope in this step, they will uncoat that envelope. The next step is replication. So overall, this whole process is called replication, all 7 of these steps, but also there is an individual step called replication. And that, obviously, the way it sounds, it is where the virus will, they will replicate their genome. The next step is assembly. So in this step, during replication, they will replicate their genomes, but also the virus will replicate, they will make proteins. And so in assembly, these proteins will form a nucleocapsid around the new genetic material. So let's put that here. Form nucleocapsid. And this basically just protects the genetic material so that when, sorry, I can't really talk and write at the same time, around genetic material. So, like I was saying, this just protects the genetic material so that when the virus goes to leave the cell, it, all their, DNA or RNA doesn't go everywhere. The next step after assembly would be maturation. Let's hope I left myself enough room. And maturation is when the virus becomes infectious and it is released from the cell after that. So, like I just said, the last step is called release. So, all in all, you have attachment, penetration, uncoating, replication, assembly, maturation, and release. What we'll be talking about in this video today is this step specifically, the replication step. So I'm going to zoom out over here, and we are going to talk about, the Baltimore classification system. So I'll write that out. This system is a way to classify viruses based on their genome. So other systems might classify them based on maybe their specific genes or what they have in common with other viruses, or maybe based on the type of host they infect. But this system is based on their genome. So there are 7 steps. I'm going to zoom in because we got a lot of information here, and I don't want to run into that. So there are 7 different types of genomes in the viral world. So 1, we have just your basic double-stranded DNA, easy peasy. Then we have your single-stranded DNA. And we're going to go in depth on each of these because each of these has a different replication process that it goes through to make fully infectious viruses, or virions, which is the singular of virus. So moving on, we have double-stranded RNA. we have, okay, here's something that you probably haven't seen before. So there is a positive sense RNA and a negative sense RNA. They're both single-stranded, but we put them in different categories. So 5 is negative sense single-stranded RNA. 6. Also, something we haven't seen before is viruses that reverse transcribe. They're also called retroviruses. And so we have 2 different types of those. We have RNA retrovirus, and I bet you can guess the other one. It is a DNA retrovirus. All right, and I will just make a note here. Let's see. Reverse transcribe. All right, we are going to go ahead and dive in to the first classification, which is double-stranded DNA. And I'm going to zoom in down here to give us a lot of space, make sure we're not running into anything. All right, so we have class, let's have a fun color. Here we go. We have class 1, double-stranded DNA. Sweet. Okay, so I'm going to just go ahead and draw out this process, and then when I'm done with that, we can go back and talk about each of these. So inside the virion, which again is the structure of the individual infectious virus, it's called a virion, that contains double-stranded DNA genome. And obviously, to get a genome for the final virion that's going to be released from the cell, you have to replicate. I'm going to try to write sideways here, which is probably not going to go well. All right, you have to replicate and to get another double-stranded DNA genome. I'm trying to combine words here. But also, like we talked about earlier, you have to have a nucleocapsid to surround that genome. So to make proteins, you have to first go through transcription, which is going to give you the mRNA. Then translation. See how small I can write here. And that is going to give you proteins. Oops. So then those 2 things put together, like we said up here, we have assembly right there. Those things are going to, okay, if I can zoom in again, those things are going to assemble into the completed structure. So let's go back and talk about these. All right, so we're going to go back and talk about each step in this process. So viruses with double-stranded DNA genomes follow the same replication process as in humans. So you have your virus, and it is going to go up to a cell nucleus and go in here for replication. So the viral DNA will enter the nucleus of the cell, and it is going to utilize the host's replication machinery, like host DNA polymerase, which is abbreviated DNA pol, and host RNA polymerase 2, which is also abbreviated pol, and then we put a 2. So the reason why viruses don't bring their own DNA polymerase, RNA polymerase, as well as other things that help them with replication is it would just make them too big. So how viruses get so small is they just don't have very many genes. Really, their genes encode things like their capsid proteins rather than all of these things, DNA polymerase, RNA polymerase. So once they get into the cell nucleus, they are going to use those things for replication. The transcription of the original genome into messenger RNA occurs first, and RNA polymerase is what helps with that. So this helps with transcription. I can't spell today. Whereas DNA pol helps with again, can't spell. Replication. Alright, so this transcribes into, forgot to say that, mRNA, which is this step right here. Alright, so after transcription, we have translation. And this is facilitated by the ribosomes of the cell. And the ribosomes of the cell, along with them, the tRNAs will help, or specifically the tRNAs will help with transporting amino acids. So we'll transport, and this is how the viral DNA will go from, here we go, we'll go from replication here to transcription to translation.